Welcome to this video on direct leukocyte single-cell type transcript abundance, or LSTA assays. This kit allows for obtaining single-cell type gene expression directly from a blood sample using probes and primers. Assays are currently available for monocytes, neutrophils, and B cells. We have optimized TACMON probes and primers to measure monocyte expression of two ratio-based biomarkers, VNN1 to PSAP for bacterial infection, and IFI27 to PSAP for viral infection, or SLE. While two-step digital RT-PCR is preferred, this method also works with qPCR. Here are example results from dPCR. In example A, high IFI27 expression indicates interferon activation associated with viral infections, SLE, or interferonopathy. In example B, high VNN1 expression indicates a pro-inflammatory reaction, often seen in bacterial infections or Kawasaki disease. These ratios effectively differentiate the host response in febrile patients between interferon and pro-inflammatory pathways. Notably, the IFI27 to PSAP ratio outperforms other biomarkers in the detection of viral infections. The results of these ratio-based biomarkers are virtually identical across different brands of digital PCR machines, demonstrating excellent cross-platform compatibility. Our kits include the necessary probes and primers, and we provide calibrator samples for use in qPCR or as quality control for dPCR. The workflow follows a two-step digital RT-PCR process, starting with blood collection, followed by RNA extraction, dPCR using our probes, and finally, data processing. Results are typically available within 3 hours. We also have plans to develop a ready-to-use tube. A ready-to-use tube system for this assay is currently under development, with a patent pending. For more information on the research and validation behind these methods, please refer to these two publications by Tang et al., which detail the translation to clinical blood tests and the use of monocyte reference genes.